Welcome everyone to this week's EV Journal Club. It's a real pleasure for me to introduce our presenter today, Andreas Zietzer, who is joining us from Bonn in Germany. And so we have the topic in front of us today of microRNAs and extracellular vesicles. And we've had some very interesting journal clubs over the past 6 months that have involved microRNAs in one way or another. And one of the big questions, of course, about microRNAs and EVs is how they are loaded. Are they specifically loaded? Are they non-specifically loaded? And what mechanisms are responsible for that loading? So we're going to hear a little bit about that today. And so, Andreas, thank you so much for joining us. And I will turn the screen sharing over to you.
Thank you very much, Ken, for this nice introduction, and thank you also for giving me the opportunity to present our work here. My name is Andreas Zietzer. I'm from the University Hospital of Bonn, and I'd like to share with you some work about how the heterogeneous nuclear ribonucleoprotein U, hnRNPU, regulates the sorting of microRNA 30C into large extracellular vesicles. I would like to start with a conflict of interest slide. We have no conflicts of interest to report. And this study was funded by the medical faculty of the University of Bonn, as well as by the German Cardiac Society, the German Research Foundation, and the Corona Foundation, which has actually nothing to do with the coronavirus, but is a small foundation which focuses on vascular biology and basic research in I would like to start with some of our previous work, just to give you a little overview on why we came up with this topic and why we got interested in this topic. We have been, as a cardiology department, we have been interested in vascular biology for a long time and have worked almost exclusively with large extracellular vesicles, formerly called microvesicles, and before that, microparticles. Nevertheless, and we use those vesicles from endothelial cells, and we found that microRNAs are packaged into those vesicles, for example, microRNA 126, and that this is somewhat changed under certain conditions, for example, in diabetic conditions, and what we published in 2013. And then later we could also show that microRNA 92 is also changed under certain atherosclerotic conditions in large EVs. And therefore we were just interested in how is the packaging actually working in endothelial cells in those large extracellular vesicles. Then some background on what is known about microRNA packaging into EVs in general. I have already said that EVs are means of intercellular communication, and that those EVs transfer microRNAs between cells. There are, in my opinion, 3 important types of mechanisms described for small EVs, where most of the research has actually been done in small EVs. The first one being that an RNA binding protein such as hnRNPA2B1 or SYNCRIP or the arginine protein 2 bind those microRNAs and act as some kind of a shuttle and get exported via the multivesicular endosome together with those microRNAs. So the protein binds microRNA and works as a shuttle and to export those RNAs into exosomes. Then already in 2010, another mechanism via lipids, via ceramides, has been described involving the neutral sphingomyelinases 2, SMPD3, and this has been shown to also be involved in the packaging of microRNAs in small EVs, but I think nowadays it's quite accepted that this is not really a specific mechanism of microRNA export, but more a mechanism of vesicle generation itself, because inhibitors of SMPD3 have been used to inhibit vesicle generation itself for exosomes, at least. Then Another quite interesting mechanism is the binding of microRNAs to target mRNAs. This has been discovered by Squadrito and co-workers in 2014, and they could show that the export of microRNAs is actually dependent on the abundance of target mRNAs in the cytoplasm. Meaning, the more target mRNAs there are, the more the microRNA gets bound, and the less it gets bound, and the less it gets exported. However, for large EVs, and this is what we've been working with for large EVs, only I'm aware of one particular mechanism, also involving hnRNPA2B1 and caveolin-1, which also works as a sorting mechanism. So there still is a huge gap of knowledge, which we try to address with this study. Here's our isolation protocol. It's pretty straightforward. We use human coronary artery endothelial cells from female coronary artery endothelial cells, which we purchase from Promocell, and those cells are subjected to serum-free culture for 24 hours before the EV isolation. Then we, of course, take this culture supernatant and do first a short centrifugation step to remove debris and cellular remnants, and then we pellet the large EVs at 20,000 G for 40 minutes, wash the pellet and pellet it again. Then, of course, we performed characterization experiments by immunoblotting. We found that large EV markers such as annexin and flottilin are abundantly present in those EVs. Also CD9 and CD63 are present, whereas the cytoplasmatic protein beta-actin is underrepresented in comparison to the cellular lysate. Then we performed nanoparticle tracking analysis. This is Zeta view, what we used here, and we found that the size ranges of those EVs ranges from around 70 to 600 nanometers. And the same thing was confirmed, or this was confirmed via electron microscopy, where we also found the size distribution with from 70 to 600 something nanometer. Then, because we wanted to find new proteins which could be involved in the sorting, we performed a proteomic analysis. And first place, we were happy to see that most of the proteins were annotated as vesicular proteins. And then we had a closer look at RNA binding proteins. And here we found first the ribosomal 14S S9 subunit, but also hnRNPA2B1, which has already been shown to have an important role in into microRNA export into EVs, small EVs, and also large EVs, as I said before. And then the next most abundantly expressed protein or packaged protein in those EVs was hnRNPU, which we therefore used as our candidate. Then we performed a degradation experiment. Most of you are probably familiar with this type of experiments. We first did immunoblot of untreated EVs, then of EVs treated with proteinase K, which would degrade all proteins outside of the EVs. Only the protein inside the EVs then stays intact, because the membrane of the EVs protects the hnRNPU from this proteinase. And then if you also add Triton X, which is a detergent to destroy the EVs, then the protein hnRNPU gets completely degraded. This basically shows that hnRNPU is actually inside the EVs. And to further to further see, to further have a look at what the actual function of hnRNPU for microRNA export, we performed siRNA mediated downregulation of hnRNPU, which worked quite well in those human coronary artery endothelial cells. We had a remaining expression level of around 30%. And then we looked at basic cellular functions such as viability and apoptosis induction to see that our model is valid in terms of basic biological functions. And here we saw that viability was not changed much, but it was still significantly changed. It was a little bit increased upon hnRNPU knockdown, while apoptosis induction was not a problem at all at this knockdown. Then we wanted to see if hnRNPU knockdown would have an effect on the vesicle release itself, because we were interested in a sorting mechanism, not so much in a mechanism which would affect EV release. And again, NTA showed that EV release was not significantly affected by hnRNPU knockdown, and also size distribution of those EVs was similar after hnRNPU knockdown. Then we performed microRNA array analysis with a QPCR based array. It's just array cards from Thermo Fisher. I think some of you might know them. And we used those array cards to simultaneously analyze around 400 microRNAs. And this was done in cells first. And here we found that in the volcano plot that the population somehow leans to the left. You can see that almost no microRNAs are up or are increased in the expression, but most of them are reduced, while if you use the correct FDR, no microRNA is significantly downregulated. And then we had a look at the expression levels of those microRNAs in EVs. And here we found that the whole population somehow leaned to the right, where we saw that especially microRNA 20A was significantly upregulated. And then we calculated relative expression by dividing the EV expression by the cellular expression. And here we saw that, again, we saw a continuum where some microRNAs like 20A, 7D, and 30C were highly significantly upregulated or increased in export, whereas microRNA 125 was, for example, not changed at all. To confirm these findings, we performed single QPCR analysis, and here we basically found similar results. 20A, 7D, and 30C were significantly downregulated in the cells after hnRNPU knockdown. And whereas in vesicles, in those large EVs, we found that they were all upregulated by trend, with only miR30C 5P being the only significantly upregulated miR. Then, if we calculate relative export, we see again that 30C 5P is the most significantly upregulated miR, but also LAT7D and 20A seem to be more exported after hnRNPU knockdown. Of course, this could be due to, for example, a normalization artifact, and therefore we repeated this analysis and performed copy number analysis, where we use a concentration row. And here we found exactly the same result without any normalization. miR30C levels go down in HCACs upon hnRNPU knockdown. miR30 30C levels go up in the respective large EVs upon hnRNPU knockdown, and also the EV, the mere copy number per single EV is also increased. So the export is actually increased of microRNA 30C after knockdown of hnRNPU, which is not, I have to say, which is not what we had expected in the first place. If you look at the other proteins, A2B1, ARGO2, they're all acting somehow as shuttles. So you would expect if you downregulate those shuttle proteins that the export would cease or would be reduced. Okay, but we went further on with this and thought we would just turn the thing around and overexpress hnRNPU by using an overexpression plasmid. Your mRNA expression was significantly increased of hnRNPU. And we again first had a look at viability and apoptosis induction. And we saw that hnRNPU did not, overexpression did not significantly affect the viability of those endothelial cells, and apoptosis induction was only mild, and early apoptosis here is slightly but significantly increased. And then, of course, we had a look at the microRNA 30C5P expression, and here in cells we did not find much of a difference, but in EVs, the 30C5P was significantly down-regulated, and also the was less present, let's say like this, and also the export was significantly reduced. And yeah, this is then actually exactly the opposite of what we found what we found after downregulation of hnRNPU. Okay. So hnRNPU has an effect, at least on the export of microRNA 30C, and it somehow seems to negatively regulate the export in large EVs of microRNA 30C. We then tried to find out what is the interaction, what is happening here, and therefore we first performed an in silico analysis where we used the RPI-seq tool, which is a publicly available tool where you can type in just the sequence of the protein and the sequence of several RNAs, and it then gives you a prediction value from 0 to 1, which tells you how likely it is that those proteins interact. And here we found a similar distribution as we had in our microRNA array. microRNA 125A was predicted not to bind at all, whereas 20A, 30C, and 7D were predicted to bind. And we therefore did a correlation analysis of the minus log p value of the export array analysis with the prediction value from the SVM classifier. And here we saw that those two values actually correlate not very strongly, but they correlate significantly. And this means the higher the predicted binding is, or that high predicted binding correlates with significant change in the export. Of course, we then had to confirm this binding of hnRNPU and 30C5P experimentally, and therefore we performed immunoprecipitation of hnRNPU, and here you can see that RNU44 and 125A did not significantly bind to hnRNPU, but 30C5P did. This was then confirmed reversely by MicroRNA mediated pull down of hnRNPU. Therefore, we used biotin labeled microRNAs, and we see here that hnRNPU is significantly pulled down or is visibly pulled down by miRNA 30C5P and not by 125A. Then we tried to, sorry, short zip. And then, if you know the other articles with hnRNPA2B1 and SynCrypt, you know that there are always motifs where the microRNA binds the RNA binding protein, and we therefore also performed a motif analysis to see where exactly the microRNA is bound by hnRNPU. And for this purpose, we used again our array data, and we used those microRNAs as input, which were significantly upregulated in export by using the simple p-value, not the FDR. Those microRNAs we used as input, and among those we found with the meme suite tool that the motif AA and so on is significantly enriched among those RNAs. And again, we confirmed this experimentally by an electrophoretic mobility shift assay, where we used miRNA 30C5P and mutated versions of miRNA 30C5P. Here in the first line, you can see the native 30C5P and how it binds hnRNPU. Then in line 3 and 4, you see the mutated 30C with a simple mutation where the purine and pyrimidine structure is preserved. Here you can see that still the binding is intact. And then we used another mutation where purines and pyrimidines were completely exchanged, and suddenly the binding is completely abolished, which is also true for miRNA 125A, always on negative control. And you can see that it also does not, or it does only bind, that it only binds very to very few extent. Okay. Then we try to find out what is actually happening in the cell to the microRNA. Okay, maybe a short summary here before I go on. So what we know at this stage is that hnRNPU affects the binding, the export of miRNA 30C in a negative way. More hnRNPU leads to less export of 30C in EVs. We know that miRNA 30C is directly bound by hnRNPU, and that this happens in a sequence specific manner with a motif which we confirmed here. Then we wanted to see what is actually happening inside the cell, and therefore how do those proteins interact, and what is the function of hnRNPU. And therefore we blocked the regeneration of of all microRNAs basically, and used actinomycin D treatment, and combined this with hnRNPU knockdown to see if miRNA 30C was maybe faster degraded in the absence of hnRNPU. Here you can just see that the actinomycin D treatment was effective. The unstable c-Myc mRNA is rapidly degraded. Then here we can see 16 hours of actinomycin D treatment in presence and absence of hnRNPU. In absence, in presence, the upper line, 30C5P, is stable over all the time, almost completely stable. And after knockout and knockdown of hnRNPU, the miRNA 30C gets degraded rapidly. And here we compare this on the right side with 125, which also is a little bit affected, but much less than 30C5P. We know from previous reports that hnRNPU is a mainly nuclearly expressed protein. This was confirmed here by immunocytochemistry. hnRNPU is green and DAPI blue, and you see that those nicely overlap. And to see if this how this affects, how the nuclear abundance of hnRNPU affects the distribution of 30C in different cellular compartments, we performed separate analysis of nuclear and cytoplasmic RNA, also in presence and absence, or after downregulation, it's better than absence, to downregulation of hnRNPU. And here we saw that the nuclear portion in the control group, the nuclear portion of microRNA 30C is higher than the cytoplasmic portion. Then if we knock down hnRNPU, this gets reversed. The nuclear portion is then now smaller than the cytoplasmic portion. And this was confirmed via MIRFISH, where we used probes against 30C and 125. Here on the left side, in the upper panel, you can see that in control conditions, 30C is almost exclusively in the nucleus, whereas after knockdown, it is in also. I'm sorry, I have to move something here. Now, the nuclear signal faints, and the cytoplasmic, there's also now some signal in the cytoplasmic area. If you compare this to a microRNA 125, there's almost no difference in the distribution after hnRNPU knockdown. So we see that hnRNPU binds microRNA 30C, it stabilizes it in the nucleus, and if upon downregulation of hnRNPU, the microRNA gets released and somehow relocated into the cytoplasm, and then probably exported. So as a next step, we then wanted to see if this increased export of miR30C after downregulation of hnRNPU is biologically relevant, and therefore we took EVs from hnRNPU knockdown cells and incubated them with otherwise native human coronary artery endothelial cells. And first, we wanted to see if this would change anything in the miR content of those recipient cells, and we found that the treatment with those hnRNPU knockdown cells, where there's more microRNA 30C, which have more microRNA 30C in them, that this also leads to an increase in those treated cells of microRNA 30C. Then we looked at basic cellular functions. Again, migration is very important for endothelial cells, for endothelial regeneration. Here we found that endothelial migration is actually reduced upon treatment with those hnRNPU knockdown EVs, up here in the scratch assay and on scratch wound assay, and the bottom in the transfer migration. Then we performed external overexpression experiments with microRNA 30C mimic and inhibitors to see if this is plausible, because if 30C is increased and this reduces migration, this should also be the case if you externally increase microRNA 30C in the cells. And here on the right side, you can see that just transfection of miR30C mimic also reduces the cellular migration of HCACs, and the inhibitor does the opposite down here. As last experiment, we then performed overexpression of miR30C in EV donor cells, took those EVs and incubated them with native endothelial cells to mimic upregulation of 30C 5P in those EVs, and this also led to a reduction of endothelial migration in EV recipient cells. Okay, good. To sum this up, we found that hnRNP on the molecular level, we found that hnRNPU binds microRNA 30C5P in a sequence-specific manner, and that it retains the microRNA in the nucleus. By this remote binding, 30C is not available anymore for vesicular export. And if you, after downregulation of hnRNPU, the microRNA then gets liberated and is exported into large EVs and then transferred to, can be transferred to other endothelial cells where it reduces cellular migration. Good, then I would like to thank you for your attention. Thank you for your attention, and I would special thanks to Gabriel Hosen, who is my co-PI, who's my co-investigator here in this study, and also to Felix Jansen, who is my PI, and yeah, he made this all possible here. And special thanks also to Nikos Werner and Georg Nikinik, who is the head of our department. Thank you very much.
Well, thank you, Andreas. This has been really good to hear about your work, and we have a lot of questions that are coming in. I just want to say I love that picture. It's kind of like you must have set up a camera in the hood there. It really looks nice. Looks like everybody's enthused about the science. So I'm going to go ahead and allow people to unmute themselves as I call on them. Because we do have some questions. Some have come in to me privately as well. So if anybody's out there without a microphone that's working, feel free to just send the questions to me in the chat box too. So let's start here with a question from Young. So Young, I'm not sure if you can unmute yourself and ask this question about the small and the large EVs. Is Young here? Well, in any case, I will just, the question is, how do you distinguish small and large EV? So you were using centrifugation to obtain the kind of a lower speed pellet, and then you confirmed the sizes with TEM and NTA. So what have you used any other methods? Do you have plans to use any other methods to look at maybe other classes of EVs?
Yes, we have recently also started to look a little more on small EVs. Therefore, we have used ultracentrifugation with 100,000 G. Of course, I think this is like the most common approach, and we have also I've used filters to reduce or to have a little sharper size distribution for large EVs. We're like 600 nanometer filters, and yeah, but not at all in this project. So.
Okay, thanks for that. Let's see here. Pasquale, you have a question about the detection methods. Is Pasquale here? Maybe not. No, I'm not sure. I just want to make sure that we are able to unmute. It looks, yeah, I think we're able to unmute, but in any case, okay. So I'm just going to ask this question: the hnRNPU, did you detect that by Western blot? Was there some other method that you used to detect it in the large EVs?
No, it was Western blot with this degradation experiment, and also the mass spectrometry. These are the two methods we used.
So yeah, mass spec and Westerns. Okay, that.
Maybe a little comment on this. We at the beginning, of course, we thought that the vesicular abundance is the key to the mechanism here, but in fact, it's the nuclear abundance or the cellular abundance, probably the nuclear abundance. So this is, yeah, somehow a different mechanism than what we've seen with the other proteins. It's more like what I explained in the beginning with remote binding by other target mRNAs. And I think that this can also be true for other RNA binding proteins. It does not have to be exclusively the case for hnRNPU, especially for large EVs, where some people argue that they are maybe not so selectively packed by escort mechanisms as small EVs.
Good. Okay. Ravi Kumar Alori, you're next. We'll see if we can get anybody to unmute themselves here. Ravi, are you there? Okay, we'll come back. So next is Enza Ravni. Is that you, Natasha?
Yes, if I unmuted myself.
Yes. Okay, so it is working. All right, thank you.
So at least the checklist, everything is working. So first thanks, Andreas. For me, this is a learning experience, a very nice study for those that, like me, are not in the field of RNAs. And therefore, it's a question for you, but also maybe for the others. I move it a little bit to a step before. So how does this RNA binding protein finish up in the vesicles? Does it has some preferential sorting itself? So it's how these proteins are sorted into EVs, and then are they present in the majority of EVs that come out of the cell? Do you have any idea on that?
I have to say I don't know exactly how the protein is exported into those large EVs. We have looked at several other mass spectrometric analysis, and hnRNPU has been found in various types of EVs, also in small EVs, and maybe the function there is different. It could be that it still works as a shuttle in small EVs, or I cannot say, but lots of RNA binding proteins, almost all hnRNPs have been found in at least small EVs and also in large EVs. Also, I mean, microvesicles, as we used to call them, and exosomes.
And yeah, thanks. I mean, this I find it really fascinating, and then I gather it's very difficult to understand in which portion of EVs this. I mean, we are methodologically quite limited in that still. And then I wonder that I think I saw somewhere that these proteins are actually regulated also according to cell type or condition, and for example, with age or stress. And can it in some way be related to the motives of why RNAs are sorted into EVs and secreted out of the cells? It's like stress responses or something like that. Do you have any idea on that? Whether this is affected by a cell type or a condition?
Cell type, yes, we looked at this. I didn't include it into, it's actually in the manuscript, but I didn't include it into this presentation because I thought it would be too long, but actually it was so fast, I could have easily done it. So it's here. We checked with fibroblasts and THP1 cells, and in fibroblasts, the mechanism works actually similar. We also see an increase in vesicle levels of 30C, and after hnRNPU knockdown, with THP1 cells, it didn't work, and there's no difference actually. And we also did separate RNA isolation of nuclear and cytoplasmatic fractions from THP1 cells, and here we also didn't find this this shift. And yeah, we speculated that this might be due to the large nucleus and the relatively small cytoplasm, or to the fact that the THP1 cells are non-adherent and the shifts between nucleus and cytoplasm are also different. But finally, I have to say we don't know yet. And then to your second question, whether this, or to the other question in any case, whether this could be relevant for any disease, or this is what you mean, no?
Yeah, condition, stress, even disease, yeah, aging.
Yeah. We are working on a second project where we found that that hnRNPU might be relevant in kidney disease, but I'm sorry, I cannot tell you more. This is still under revision, but we think that there can be that this is relevant.
No, thanks. I will read it when it's out. Okay, thanks.
Very good. Thanks for the question. So we have a few questions that relate to the subcellular localization of hnRNPU. And so one of those is from Ursula Sandow. Another is from Mona Batish. So I'd just like to ask both of you if you want to unmute yourselves and discuss your questions a little bit with Andreas here. Let's see if we can find out a little bit more about nuclear localization and what that means.
Sure. Hi, can you hear me? This is Ursula.
Hi, Ursula.
Yeah, so my question was, since you saw it sequestered in the nucleus, is it affecting the processing of the microRNAs, or is your assay able to distinguish between the immature and the mature forms of the microRNA?
This was actually also asked by the reviewers, and this is an excellent question. The FISH assay is not able. We checked this also with Thermo Fisher, who made the probes, the FISH probes. The FISH assay is not able to distinguish between cellular and between pre or mature mirs, but we also performed an analysis of pre-mir 30C1 and pre-mir 30C2, and they were not at all changed after hnRNPU knockdown. So and also I think pre-mirs, we checked this. The pre-mirs are much less abundant, so I think most of the signal is still from mature mirs and not from pre-mirs, and the QPCR array, the Techman arrays we used here for this QPCR analysis, they are specific to mature mirs. So I think we kind of excluded that this is all pre or premier related.
So this is Mona. So basically, does this microRNA in its mature form, is it supposed to function in the nucleus biologically? I mean, what is its biological role? What does it do?
Yeah, in the cell, in the clear cells, for example, as we showed later, it reduces migration, and I think several microRNAs can function in the nucleus and outside of the nucleus. So, but
because I'm just trying to understand, because what I get from it, maybe I didn't follow it complete, because your main central message is that hnRNPU is going to sequester this mature RNA in the nucleus.
Yeah.
Right? And so what is the biological role there? Like, what will happen if it doesn't do that? You know what I'm saying? And follow up on the other question. I mean, there is a way you could do, regular fish for the pre-microRNA, which is longer, and you can do regular fish probes for that, and then see if that localization changes. as compared to the mature RNA, and then you can see if it is actually affecting the processing of the microRNA, or is it just sequestering the mature microRNA in the nucleus?
Yeah, I mean, the processing, I think this we somehow excluded because the RNA, the levels of the premiers were not changed. So if this would hamper processing, then the premiers would at least have to go up in the same amount as the 30C, the mature microRNA is reduced. Then regarding the biological function, I'm not entirely sure what all functions are, but it is mainly expressed in the nucleus. There are other studies who show that the 30C is highly abundant in the cellular nucleus.
Okay, thank you.
And I have one more like kind of related question to all of this. So if the miR is bound to the RNPU, is it still have ability to bind and interact with targets? Or is it sequestered also from its biological function as well? Like, is it inactive in an inactive form?
I have to say we don't know.
Okay.
Yeah, because we checked some basic cellular functions like viability and apoptosis induction. We saw some effects, especially for viability. I don't think that this is just chance or coincidence. I think there's certainly some effect, and hnRNPU the knockdown of hnRNPU will have also in the mother cell will also affect biological functions of the mother cell, and of course this the stabilization and all of this will hamper the role of 30C and also of other microRNAs. Does this answer your question somehow?
So, yeah, thank you.
All right, so we have a question about specificity from Roger.
Can take over if my microphone's no good, but I'm
Okay.
wondering if you looked at other microRNAs with binding sequence similar to 30C5P, or if you did, like exploratory RNA-seq to look at altered regulation of other classes of RNA.
No, for this project we didn't do any further than what I have here. I mean, it could be that there are other motives, and but yeah, no, we didn't do RNA-seq, but yeah, it could be that there are other motives, and it could also be that there are other microRNAs involved, because obviously there are much more microRNAs than we tested here, and yeah, it could be.
So those are possible candidates, but you didn't check their levels in response to alteration in the protein level?
No, in depth we only checked 20A, 30C, 70, 125A, RNU44, of course. Yeah.
And I might have missed this in your fluorescent images. Did you show that when the RNP in EVs goes into nuclei of the target cells, or not yet?
No, this we didn't show. We didn't follow up on the protein transfer, but only on microRNA transfer. So no protein analysis was done in target cells.
Thanks. This is very thorough.
So Roger, I think you had another question too about whether there could be free complexes. Is that right?
Yes, I'm always looking for RNAs bound to proteins in free circulation. I wonder thoughts on that for this protein, like actually not sequestered in EVs but released free in circulation.
I'm not aware that hnRNPU is freely circulating, but in our case we did this degradation experiment to see that hnRNPU is actually inside the vesicles, but yeah.
Yeah,
I
that's
don't
an
know.
interesting question, because I suppose, you know, if we're talking about biological fluids, you know, if there are cells that are dying, breaking open, you
Yeah,
could certainly get some of that there. But, you know, in cell culture, maybe if you have, you know, very healthy cells, you might not see that. So it's maybe this is another good candidate, Roger, for carrying RNAs outside the cell. Good. Okay. Let me see here. We have a question from Peter. Although Peter, it appears that Peter has left the call. So the question though is the following. So RNPU is known to interact with glucocorticoid receptors. Can you think of endocrine conditions that could alter EV cargoing with miRs? Peter asks.
Yeah, I already told you one, which we are working on. So I cannot really go further into detail here. So, but yeah, of course, also glucocorticoid receptors, and we didn't, for example, we didn't look at all on post-translational modifications here, on other interaction partners, on other ways to change the conformation of hnRNPU. And of course, this could all affect the binding to microRNAs.
Yeah, so it seems like a plausible mechanism for some of this sorting. So that's quite interesting. Our next question here, I'd like to call on Bethany, who asks about the consequences of the knockdown. Bethany.
Hi, thank you for such a great talk. This is a very interesting area of research that we've been working on too, as far as RNA binding protein loading, microRNAs, and EVs. So yeah, my question was just about when you knock down hnRNPU, are you just allowing for binding of other RNA binding proteins to come in and bind with MC and then transfer to the EV?
I'm sorry, there was some acoustic problem, but as I understood, your question is if other RNA binding proteins somehow take over if hnRNPU is knocked down.
Correct. Yes.
Yeah. Again, I have to say we didn't further look into this yet, but it could be. I guess it's very likely because our microRNAs are, or almost all RNAs are, rarely just naked by themselves in the cell, but most of them are always protein bound. And I think this applies also here as well. If one protein is downregulated, they might bind to other proteins. Yeah.
Okay, let's see. We have I think we've had several questions that have come in the chat box that probably have been largely answered, I think, by your responses to some other some previous questions. But I'd oh, and it looks like Marco may have left as well. But Marco Licalzi has a nice question here, and I think it prompts a little bit of speculation. So earlier this year, Albanese et al. Presented a paper where they mentioned something along the lines of microRNAs not having a big role in communication since they're at extremely low counts in EVs. And so we had indeed had a debate session several months ago where Manuel and Michiel Pechtel discussed their views on microRNAs. So what do you think is the role of the system you're describing, asks Marco. What are the effects of miR30C5P on other cells? And I would just add to that, do you think that there's something special about large EVs that maybe promotes this kind of communication more than small EVs might?
Yeah, this is interesting, and we had a lot of discussion with the reviewers about this. actually reviews. We had excellent reviews. I think we had here. I really have to say this. And I'm going back to this copy number calculation here. You see now on the right bottom panel shows how many microRNAs, microRNA 30C copies are actually in one of the EVs. We calculated this with RNA content and NTA. And here we see that especially that the levels are quite high if you compare to exosomes, where there's normally one or less than one microRNA at all per EV, but not a specific microRNA, but of all microRNAs. There's only about there's not only one or two or per EV, and this is, in my opinion, an important difference to the small EVs here, that the levels per EV are higher, and this may also have to do something with geometry, because if the diameter is only twofold higher, then the volume is already to the fourth potential is higher. Then it's much higher already then, and the system has a biological relevance. I think we show that quite nicely at the end of this presentation, that the EV transfer is relevant in terms of microRNA numbers and also in terms of biological functions such as migration here in this case. And yeah, microRNA 30C has been implicated in endothelial healing in previous studies, for example, by Rainier Boone, who did this also years ago. So it has a function in endothelial regeneration, and also probably in other cells, but I'm only aware of the endothelial function.
Yeah, I mean, so I think Esther Nelta-Thun is on the call, but I know already back in 2012, she had reported that, at least in the systems that she was looking at, and I think it's been verified numerous times since then, microRNAs in the smaller EVs, they're actually at lower abundance than they are in the cell, and I think there have been reports that suggest otherwise, but it makes sense, because of, as you say, the physics of this. You've only got so much cytoplasmic material that can get into a small EV. But it could be, to me, I think the large EVs are very interesting simply because of their volume and all the contents that they can pack in there. So we have, I think there have been a few questions that have come in the box, and that also have been sent to me privately, that ask about what this all means downstream. So are there targets of miR30C that you find in the EVs, or targets that are in a recipient cell? So there's a suggestion from Mona that it would be interesting to look at those targets. And then we also have a question about homologs. So hnRNPU, have you looked at its conservation across species? How far can we go afield? I mean, is this a mammalian thing, or how many, is this found in a lot of different organisms?
In mammalian cells, definitely. And okay, just some comments on hnRNPU, on the biological function. It also is involved in mRNA processing, and has is absolutely necessary actually for mRNA processing. And this leads to the fact that if you completely knock out hnRNPU in mice, those mice are not, they cannot live, because this mutation is lethal in the embryonic stage. So hnRNPU is, at least in mammalians, is conserved and has multiple functions. And I'm sorry, what was the other part of the question?
I guess about just looking for the targets.
Yeah.
Yeah.
Yeah, well, this is what we usually did in our other studies, and here we try to for 126 and also for 92A, and here we try to focus a little more on the donor cell, and therefore we didn't do this, but of course it would be possible, and it just was not really in the focus of the study, I think, because we wanted to see how is the export regulated. And of course, 30C has functions. I think this was also the question from Mona Bedish. Of course, 30C has a lot of functions, but this was not the primary goal. We just wanted to show that the transfer is biologically relevant and that it does something which is important for endothelial cells and affect migration in this case.
Very good. Well, I think we're going to have to leave it at that for today. We've come to the end of our hour, and I really appreciate your sharing your work with us, Andreas. I think it's been very illuminating and gives us some more, another important protein to think about in EV-mediated microRNA communication. So thank you again, Andreas. Thanks to everybody who's joined the call today, and for all the good comments. I'm going to be sharing the text file with the comments with Andreas in case there's any follow-up that he'd like to do. And I'd just like to remind everybody that we are putting the presentations on YouTube. So we have a YouTube channel. Just look up Extracellular Vesicle Club. And you can subscribe to that channel, and you'll be able to see the presentations and also get updates of when they become available. So I try to edit things soon after the journal clubs, but it sometimes takes me a few days. So thanks again, everybody. Hope you have a great rest of the week, and I'll look forward to talking with you again soon. Bye now.